Hello, everybody. How are you doing out there? Good to see a few of you in there in the chat. I'm just double checking if I have any messages. No. Okay. I'm glad to see that we're getting a increased population in the app or in the Hangouts. I think maybe even tomorrow we could do a little bit of a hangout instead of a class. Maybe that might be the right day to do this. So I'll put a mention of that in the chat there. Okay, so yesterday we covered Mendel's principles. So Mendel had 4 principles that are important. Number 1, all traits of an organism are controlled by a pair of genes. All genes come in 2 types, a dominant and a recessive type. All of the alleles are randomized and randomly assorted to the various offspring. And if you had more than one trait, those genes are also randomly assorted. So that's really the main concepts that we are trying to get at in the previous example. So those are the 4 principles. So one of the tools that we're going to use a lot is what's known as a Punnett square. And the Punnett square is a mathematical tool, really. It's developed from a sort of math perspective, and it's a math tool to show that show those principles of segregation and independent assortment. So as a math tool used to represent the probabilities of different allele combinations possible from the cross of 2 parent genotypes. Gene according to the law of segregation and independent assortment. And I guess genes and pairs, well, allele pairs, allele combinations. So in the Punnett square, we get the 2 parent genotypes, which will be a pair of genes, a pair of alleles for one trait. We take that pair of alleles for one trait from that parent, and we are going to cross it with the other parent. And to do this, we are going to place one parent's alleles across the top of the Punnett square and one pair of alleles along the column on the left of the Punnett square. For a Punnett square, you're going to need as many rows and columns as you have alleles, essentially. So if you have 2 alleles, then you need 2 columns and 2 rows. And we will get the prediction for how many offspring are possible or likely for a given genotype combination or a given allele combination. So let's look at this example here. Let's start with these. Well, let's look at these couple of examples we have here. So here we have a heterozygous female, and a heterozygous female or male. Male is debatable here. It's a plant. So anyways, we have a heterozygous male and a heterozygous female cells. So this is a pollen grain and an ovum. And in those cells, we see we see the dominant species, the dominant gene, which is represented by capital R, probably for red. So let's use red. And if that capital R is present, then those plants are red. But these are both heterozygote plants, so that means that they are also carriers for the recessive genotype. So when these two plants cross, we end up with a couple of different options. We could get one capital R from both parents, one dominant gene from both parents. We could also get one dominant gene from one parent and a recessive gene from the other. We could also get one dominant gene from the other parent and a recessive gene from the other, or we could get two recessive genes. So maybe I'll give some grays here. And so the Punnett square is helpful because it allows us to organize this cross. We take whatever is at the top of the column and we place it down into the box. And what is ever to the left of the row, we place it in the other position in that box. For the next square, we're going to take one from the top and one from the left, one from the left, one from the top, one from the left, one from the top. And so this is a way of getting every single possible combination. There are other ways, but this is usually considered the easiest one to visualize. And in this way, we have assembled all four options. Notice that in this dihybrid cross, or in this monohybrid cross, so both of the parents were hybrids, we end up with two plants that have the heterozygote condition, just like their parents. So these are two red plants. And we have one plant that is white. And this plant will be red as well. So when we go back and look at these plants, we notice that of their children, there is a 3-to-1 ratio of red to white. Okay, so let's play around with a couple other examples. It will actually not really matter too much about what letters we use. That's just window dressing really. There are only actually a couple of variations to this question that we can even do. You could arguably memorize all of the possible ways that this question could be done and be good enough to go. So here we have a heterozygote that has the tall condition, and that is being crossed with a short short plant here. So when in this cross, this short parent has two recessive genes to donate, and this tall parent has one recessive gene, and one dominant gene. So when they get crossed, we move each of those genes down. So here I'm going to move this one right to here, and I'm going to move from the left over. I'm going to move a tall gene over into that first box. Into the second box, I'm going to take a tall gene and move it over, and I'm going to take a short gene from the top and move it over. And then I'm going to repeat the process, but I'm going to be moving only short genes now because I'm moving a short gene here, a short gene there, a short gene here, and a short gene there. And the end result is a new ratio that we haven't seen before, which is 50% or 50-50 or 1-to-1 is probably a better ratio, a reduced ratio. We have 1-to-1 tall to short. We do have a difference in genetic ratios. We have, or well, not in this case, but we have one tall, one heterozygote, to one homozygous recessive. Okay. So if we were given a question and asked to do it, let's look at this question here with this example right here. We are given two heterozygotes or two carrier green plants to cross. So what I'm going to do is I'm going to set up first by putting a little box. Oops, that's a circle. How about we try a box? Okay. And I'm going to make my box of 4 by 4. I'm going to put one parent along the top, and I'm going to put one parent along the left. So this is parent 1, this is parent 2, and they're being crossed. I'm then going to take my parents very carefully. I'm going to take the green here, and I'm going to take a green here, and put them in the box. I'm going to repeat the process. You can even do all of your rows and all of your columns all at once if you like. That's an approach that I've seen done. So someone would do all of this row, all of this column, and then they might go in and fill in the other details. Let's see, I'm going to use an orange so it's visible. And so we end up with all of our 3 combinations. Notice that this is what we call a monohybrid cross. And the monohybrid cross, or one trait cross, always has the same ratio. It has a 1 to 2 to 1 genotype ratio, and a 3 to 1 phenotype ratio. At least in the case of a Mendelian system. Oh, there's the words. So you can see the monohybrid cross explained there just below. A cross of 2 heterozygous individuals for one trait, and the result is always 1 to 2 to 1. So this, in this case, we would say 1A, one heterozygote, 2 heterozygotes to 1 homozygous recessive. A true breeding plant is a true breeding is a dependable organism that will always breed. So true breeding is are homozygous organisms that always produce the same type of trait due to the homozygous genotype. I think we can actually go farther. Homozygous parents that always produce and a purebred is our offspring bred from a true breeding pair such that they retain the same traits as their parent. Minimal difference. Now, true breeding or pure breeding of an animal, these are very similar concepts. So true breeding is sort of like from the parent perspective. Purebred is like the offspring perspective, but it's on the same idea. So pure breeding is problematic. Usually the easiest way to pure breed is to take 2 organisms that are very closely related. And in this case, you don't introduce very much genetic change in the organism, which means that it's not genetically diverse. And we know that low genetic diversity increases its susceptibility to its environment. So it's under a higher pressure, a higher genetic stress, or higher evolutionary stress, because it does not have the chance to have the adaptations needed to survive. So complete dominance is the type of dominance we see. The type of allele interaction that we're seeing in these Mendelian systems. So complete dominance is another name for the Mendelian, is also known as Mendelian, Mendelian behavior, Mendelian pattern. So this is the idea an interaction between alleles where one allele appears to completely determine the trait when present. We will contrast complete dominance in a little bit with another example, with a couple of other examples, such as codominance and incomplete dominance, where one allele will not completely determine the trait. So here the trait is effective. So in some examples of animals, for example, the dominant allele for hair color, the brown, is usually referred to as like a brown allele, and that allele will dominate over recessive alleles, so that it'll mask the behavior or mask the effect of a recessive allele, even if that recessive allele is present. So that's definitely the case that we've been discussing up until this point. There's a little bit of repetition in this example, in this booklet. Okay, so example number 1, the monohybrid cross. If you have the allele for tall plants is capital T, and the allele for the short plants could be represented with the small t, a heterozygous tall plant is crossed with a short plant. What are the genotypes of the parents? And I believe this is actually the example that we have right here. So here we have a heterozygous plant. So here's the heterozygous parent. Heterozygous tall plant is crossed with a, let's just go short plant here, short pea plant. I feel like those colors are backwards. Again, we move the T's down, we move and over, move the T's down and over, down and over, and over and down. Doesn't matter what order, that's all the same. So we can see that, what are the genotypes of the parents here? Well, the parents were a heterozygous and homozygous recessive. Notice they don't have to tell you that it's homozygous, they just have to tell you that it's short. Because by telling you it's short, it's telling you that it has the recessive genotype. It has the homozygous recessive genotype, because that's the only genotype that will produce a short plant due to the complete dominance. What are the genotypes and phenotypes of the offspring? Well, the genotypes we can see from our square, 1, 2, 3, 4 genotypes, but 2 of them are the same. So we have the TT, we get 1 TT, to 2 heterozygous, to 1 homozygous recessive. And what are the phenotypes? We get 3 who are tall compared to the 1 that is short. Okay, and so this is all just working through what we just did. Blah, 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 blah. Phenotype ratio is not this. Oh, yeah, it is. Okay. Oh, what am I doing? Yeah, whoops. Get out of here. That's not true. I did a monohybrid cross. I should just read my notes, eh? 2, and that's too short, too tall. Now, a better way of drawing either of these is to reduce the ratio. So I would expect 1 heterozygote to 1 homozygous recessive. 1 tall to 1 short. 3 hours of teaching straight in a row is a long time to be just talking straight at the camera with nobody there. So the only thing to entertain me is my small little image of myself. Okay, so the 2 here that are tall and the 2 here are short. Tall and short. Okay, and you can see that if you didn't catch that, it's all right there. All right. Boom. Okay, I will be posting an assignment related to these Punnett squares, especially these simple ones. I'll get an assignment out to you ASAP so you have some practice material to work with. And that'll be coming down the channel as soon as I'm done the stream, probably. The next concept I want to bring up is what is called a test cross. And so it looks like I'll be finished this stream in a second. So the test cross is a experiment. So it's an experiment where an experiment used to determine the genotype of an individual with the dominant phenotype, but an unknown genotype. This is accomplished by breeding the plant, breeding the individual with a recessive phenotype and looking at the offspring to identify any recessive. I'm looking at the, well, let's go looking at the pattern of offspring. Okay, so I'll show you how this works. If you were given a flower and you did not know if it was the dominant or the recessive phenotype, so for example, a purple flower here is the example that we're given. So with this purple flower, it could, because of the way that complete dominance works, this purple flower may be the result of a of a homozygous dominant genotype or of a heterozygous genotype. And so, if you breed this target, this is the target here, this is the one we're looking at. If you breed this flower with the recessive genotype, which has a known phenotype, recessive phenotype, which has a known genotype, then there are two possible results that will happen in the crossing. One result is that you will produce a group of seeds that all have the same color, and that would indicate that the plant, the purple plant, was donating only dominant alleles. In the other example, you could create a group of dominant and recessive alleled flowers, and that would tell you that the dominant flower was a carrier for the recessive trait. So it's a simple process. Certainly not something we can do with many animals. Animals are much more difficult to do this. Maybe rats or mice or flies or something. But it's much more applicable to plants, where there's going to be a huge amount of offspring that you can grow to really get a sense of what was the cross. So, for example, so you can see this example. You can see how the result might be all violet flowers here. So all violet flowers, or half violet and half white. And that's because only half of the genes that are being donated are the violet genes. Here, all of the genes being donated by this flower are the dominant violet genes, and on the other one, we have only half. So only half the plants inherit it. All right, and that is the test cross. I think we are going to leave it here today because I don't think there's any reason to rush. And I'm going to spend a couple minutes here getting an assignment up online for you. So keep an eye out on the classroom. And I also have my Google Hangout open. So if you want to send me any messages there, or if you want to chat with anybody there, it would be a great time to do that right now. So probably a more appropriately length stream today. And we'll talk to you tomorrow as we dig a little bit deeper into these crosses and things. Tomorrow we will dig into the dihybrid cross, which can be quite challenging. Okay, thank you. Have a great day.